Hello everyone, I'm Ramon Macias and I will be presenting my lightning talk on fetal AAV mediated silencing of variant Tubb2b. The World Health Organization estimates that approximately 6% of newborns worldwide are born with a congenital disorder, and this may be an underrepresentation as terminated pregnancies and stillbirths are not included. For a subpopulation of congenital disorders, both surgical and non-surgical options are available. Unfortunately for patients with tubulopathies, no clinical treatments have been available. Pathogenic variants in various tubulin genes have been linked to a spectrum of brain malformations. At the bottom, we can see an MRI of a healthy brain, and to the right, an MRI of a brain with a mutated Tubb2b, where you can see dysuria or abnormal folding in the brain. Finally, in virtually all cases, patients have heterozygous de novo missense mutations, and that indicates a possible dominant negative disease mechanism. In a normal scenario, microtubules interact with various motor, microtubule-associated proteins, and cytoskeletal elements. When mutations happen in various tubulins, that interaction is damaged, which can disrupt microtubule function. For neurons in particular, microtubules are important in axonal transportation, cell migration, and cell division. With these impeded, synapses can't form, cortical neurons can't migrate to their appropriate layer, and neuronal fate is perturbed. If mutations in tubulin cause such drastic phenotypes in patients, what would happen if we deleted an individual tubulin gene, specifically Tubb2b, as it is one of the most commonly mutated beta-tubulin in tubulinopathies? Humans have 9 beta-tubulins throughout the genome, and all of them have high sequence homology, as can be seen in the heat map. And so we expect that there should at least be some partial redundancy between beta-tubulins. Upon deleting Tubb2b, we saw no large differences in brain weight and no differences in brain architecture in both whole mount and histological slides. And so there is indeed at least one beta-tubulin, if not multiple beta-tubulins, that are compensating for the loss of Tubb2b. And so we're seeking to take advantage of this built-in redundancy and create an AAV vector that can delete mutated Tubb2b. To generate our AAV vector model, we have incorporated 3 main elements: a human synapse 1 promoter that targets the central nervous system, a miniature Cas9 enzyme to induce a double-strand break, and 2 single guide RNAs to bind Tubb2b and recruit our mini Cas9 to the site for deletion. To test the AAV vector, we will be conducting a functional assay, where we will deliver in vitro our vector into mutant Tubb2b primary neurons, and then quantify the rescue by using a dendrite and axon marker in neurons, MAP2, to measure the neurite length. Already, in previous experiments, we've been able to show that heterozygous Tubb2b mutations have lower neurite length, and homozygous mutations have even lower neurite length. After the AAV in vitro experiments, the future goal is to deliver the AAV vector to our mutated Tubb2b mouse model at embryonic day 12.5, which is the start of neurogenesis and is before any cortical malformations can develop. We'd then like to see if they survive at normal Mendelian ratios, see if they develop any cortical malformations at peak radial migration and at the end of neurogenesis. And then lastly, we'd like to see if they have any behavioral issues.